Sri Gurubhyo Namaha. Welcome to our online teaching academy, and I welcome you all for the biology class. This is class 12 biology, and we are in the chapter evolution and genetics. In genetics, already we have completed one topic. Okay, that we are going to have a quick recap. So let us have a quick recap. Not from here, recap. So we'll see. We started with Gregor J. Mendel, who is the father of genetics. And what he proposed, he told about the inheritance patterns, how the characters are transmitted from one generation to another generation. Next comes T.H. Morgan, and he was explaining about the linkage and crossing over. Next, the scientist was saying about the vehicle. Something acts like a vehicle and it carries a gene. So it's nothing but chromosome. Who gave the theory, chromosomal basis of inheritance? Can you tell me? Yeah, it is Sutton and Bower. So several scientists with several proposals, they were explaining about the inheritance patterns. So totally the lesson was about principles of inheritance and variation. Now we are going to go deep into the molecular level. So molecule, when I say gene, we are saying gene, you can see the cell is showing the nucleus and then something like a thread-like structure coming out. What is that? That is the DNA. So in DNA, something like a bead, some arrangement, gene. So we are going to go in deep, the molecular level. So I would like you to travel into the molecular level. So we'll move to the molecular level now. The lesson topic is molecular basis of inheritance. And here we are going to deal totally with the world of DNA and RNA. So first, if anything you take, it should have a property, characteristics we can say. We do have character. Like that, here also when we say genetic material, it should have a basic character. So what is a basic character? We'll see one after the other. Characteristics of genetic material. So first, it should replicate and it should be stable and inheritable. It should replicate, it should be stable and inheritable. What is inheritable? It should transmit the character from one generation to the next generation. Like this, it just goes on. And then it should have mutation property. Mutation actually is lethal, but it also has advantage also. Okay? Then it should able to translate and change the information, transcribe. So I'll say once again, what is this translate and transcribe? This is a very important thing, and the whole lesson, majority of the lesson, we are going to deal with this property. So something is going to copy. You can see something going around and around. It's nothing but the 3D, the dimensional of DNA. And this DNA going to copy the message and it going to transcribe, okay, that we call it as copying by mRNA, and it goes to the next one, protein formation. That we call it as translation. So this is translate and this is translation. Okay, so both translate and transcribe the message. Okay, so we are going to see this one after the other. So we'll move to this structure first of all. So when I say DNA, just a double helical structure we are saying. It's just a double helical structure. So what is the structure made up of? It has a small unit, and we are going to see this unit in detail. When you say nucleic acid, there are two nucleic acids. One is DNA, another one RNA. So we'll see one after the other, the nucleic acid. There are two nucleic acids, DNA and RNA. And this is another important one mark question. Please make a note. Friedrich Miescher, who gave the term nuclein, and from where he saw this nuclein? In pus cell. Okay, this is a one mark question, board based question. And next, if you take, who coined the term nucleic acid. Mr. Altman, he coined the term nucleic acid. So, in nucleic acid, what it is composed of? What is nucleic acid actually? The nucleic acid is nothing but a polynucleotide. It's like a chain of formation of a different things. Okay, what are the different things? 3 different things we are going to see. So, we will see one after the other. So, I think this is familiar for you. You have read in 11th standard in biomolecules, right? It is a polymer of nucleotide. Okay, what is polymer? Something like a chain. So, one after the other it is going to form, right? It consists of pentose sugar. Okay, you can see that pentose sugar, pentose 5 carbon. So, 2 sugar molecule, deoxyribose, another one ribose sugar. Is there any variation in deoxyribose and ribose? Can you figure out what is the variation? I have highlighted here also. Yes, it has 2 functional groups. Just look out where is the 2 functional group in ribose sugar or pentose sugar. In ribose sugar there are 2 functional groups, and in pentose sugar only 1 functional group. This is a major difference and very important difference. Please make a note. And next, phosphoric acid. So, one by one we are seeing. First, pentose sugar. I show the difference. One is deoxyribose, another one ribose. Okay. Next, what we are going to see? Nitrogenous bases. So, how the basis is going to be, that we are going to have a look. And then the phosphoric acid, where it is going to be attached. We will move to the next, nitrogenous bases. In nitrogenous bases you can see there something 2 cyclic structure you can see there. One is dicyclic, another one monocyclic. So, what is monocyclic and what is dicyclic? Just look down, what is the name? It is purine and this is pyrimidine. Purine is 2 ring structure, whereas pyrimidine single ring structure. Okay. And this nitrogenous base is very important. Another 1 mark question. Okay, differences between base question. So, what it has purine? It has A and G. What is A? Adenine and G? Guanine. And it is dicyclic. Again make a note, it is a 2 mark difference based question. It has 4 nitrogen. Where are the 4 nitrogen placed? Have a look. In which one? In a dicyclic. Where are the 4 nitrogen? You can see the number. I will just encircle it so that it will be clear for you. Yes, look at the number 1 and then 3 and then 7 and then 9. Okay. So, these are all the places. The same way you can figure out where are the nitrogen in this, number 3 and number 1. Okay. And this is pyrimidine and it is monocyclic. Okay. And what are these 3 letters? C, T and U. A and G, adenine and guanine. C, T and U, cytosine, thiamine or uracil. Where is the uracil? This uracil is seen in RNA, not in DNA. Okay. So, what are we saw now? We saw nitrogenous bases. Before that, what we saw about the ribose sugar, pentose, that is deoxyribose, another one ribose sugar. And now the phosphate, where it is attached, that we are going to see the nucleotide structure. So, this is the basic structure, nucleotide structure. So, number 1, nitrogenous bases. Number 2, it is what? Nitrogenous bases, then pentose sugar and then phosphate group. And this 3, how it is going to form like a chain? Actually 2 strand, we say DNA double strand, double helical structure, right. So, first that 1 strand, how it is going to form? That we are going to see. Before that, this is a very important thing, a difference between nucleoside and nucleotide. Something is absent, that you call it as nucleoside, and is present with the 3, that you call it as nucleotide. So, have a look, nucleoside, something is absent. So, what is absent here? something is absent. So what is absent here? The phosphate group is absent, and this you call it as nucleoside. With the phosphate group, you call it as nucleotide. It is another important 1-mark question. Make a note. And now we'll see the structure, the nucleotide structure, how the nucleotide will be in a single structure, and the bond formation, okay, the bond formation between the two strands. I said purine and pyrimidine, and how are they going to make a bond? A will pair with the T with double bond. C will pair with the G with triple bond. And what is that bond? And that bond is hydrogen bond. You can see the bond here. This is the way of linking with the bond. Okay. So we'll move to the next. It's a skeletal structure. What we have in the NCERT textbook. Again, another picture-based question we are going to get from this. So this is a single strand, and you can see the end. This end is again very, very important. 5' end and 3' end. This is another important, again I am repeating, because you may go wrong when you locate the polarity of these two strands. One, we are saying as we go inside the lesson, we know the type of the strand. We call it as leading strand, lagging strand, like that. Okay. The polarity, how are we going to write? 5', 3', 3', 5'. I made this actually parallel, and they run anti-parallel, we are saying. Here it is very well visible, anti-parallel. That is why the polarity is opposite, right? So the same thing, skeletal structure with a polynucleotide we are going to see. Here is the structure, and it's a double-stranded polynucleotide chain. Again, it's our NCERT book picture. It's also a picture-based question. So make a note. A polynucleotide chain which is showing with the bond, H bond. What is that H bond? Hydrogen bond. So what are the two things? A with T and C with G. Okay. Next, now we are going to move into the lesson, and we are going to see our key play of this lesson. It is nothing but the double helical structure. Something, it is right-handed coil, and it's a B-form DNA. Okay, you have here this long back, right? So it is nothing but deoxyribonucleic acid. The scientists Watson and Crick, they both gave this model. There are different forms of DNA, and this is B-form DNA, and it's right-handed double helix. Again, it is a 1-mark question. Make a note. So note down the scientist's name, Watson and Crick, okay, in the year 1953. And you can see this beautifully running a DNA structure. Here, another important 1-mark question I am going to say, just make a note, which is the backbone of this nucleotide that we are going to see. It's again picture-based question. You can see something backbone, yes. Sugar phosphate, it acts as a backbone. It's a 1-mark question, okay. And this DNA, totally, how is the coil? How many base pairs are there? There are actually 10 base pairs in one turn, okay. And what is the distance between this? It's actually 20 angstrom in distance, okay. So all these things in detail we are going to see about the DNA. Now we are going to classify the DNA. We are going to describe the structure. How this DNA is looking? We know already it is made up of polynucleotide, okay. And then what is the length? What is mean by base pair? What is the distance? Okay, that we are going to see. Again, it's a model of Watson and Crick. So let us go fast about this, because already we discussed about the measurement. It's a 20 angstrom width, and then in one turn we have 10 base pairs. When I say base pair, it is nothing but the pairing of AT and CG. Okay. Let me show the points and I will highlight the points, make a note. Yeah, it is a 3D, 3-dimensional structure, okay, and it is a helical strand proposed by Watson and Crick, okay. The polarity, and it has a phosphate group. This we discussed already. What is the polarity? It is a 3' N with a 5' N. That will be the down strand. Now I am using the word up and down. As we are going further, we will be using the term leading strand and lagging strand once we are familiarized with ourselves with the replication. Okay. Then CHARGAFF'S. We will come to CHARGAFF rule. CHARGAFF, ERWIN CHARGAFF he gave a proportion, okay, proportion about the adenine guanine, how it has to be arranged. Whatever species it is, nothing is same. Even our DNA will not be same. There will be some point differences, okay, in human DNA. Likewise, species will have a species specificity. There is a difference in their DNA. But when you take a species, that species will have a specific ratio, and that ratio was given by ERWIN CHARGAFF. So that CHARGAFF rule we are going to discuss here. Adenine, guanine, cytosine, thymine. Okay, we are going to use the same word like a mantra. Adenine, guanine, cytosine, thymine. How are they going to be arranged? Okay, so A, T is equal to 1, okay, and C by G is equal to 1. This is a very important thing, like a numerical based question we are going to get from this part. Another one, A plus T by C plus G is equal to 1, the constant, the number, right. So based on this, we are going to have a numericals. So let us see what is this. It is a grammar of biology. It is very important to find the ratio, how it is arranged in our body, this adenine and guanine. A is equal to T. Another one, just have a look. A is equal to T and C is equal to G. The number, suppose A is 260, T will also be 260. Okay. And what is purine and pyrimidine? We know already. Ag is purine, Ct is pyrimidine. Okay, children, you will have a confusion here. I am saying here At, this is a number constant. Okay, if A is 620 or 260, the number will be same. And the same thing when you come to purine and pyrimidine, there will be a question, they will ask you to find what is the purine number. Suppose if you have a pyrimidine 560, what will be the purine number? And the total number of base pair is 1000 something. By the time you have to calculate this one. Okay, this is purine and this is pyrimidine. Okay. So we'll move to the next topic. We discussed about the DNA, CHARGAFF. Next, it's a very important main theme of our lesson, and we are going to go further with this topic only. This you call it as central dogma. Okay, in central dogma, how this DNA going to prepare a protein? Again, I am saying it is not going to prepare, it is going to give a protein. How DNA is going to be constant is same there. How it is going to prepare a protein? Something need to carry the message from the DNA and it has to produce a protein. So who is that vehicle here? That vehicle is mRNA. The name itself says M, messenger RNA. Let me write. Yeah. mRNA acts like a vehicle. It goes, copy the message from the DNA and then translate the message to make the protein. So it's acting like a carrier of the message. So one we know mRNA. Another one if you take it, next comes our rRNA. So here we are going to deal totally with RNA. What is RNA? Ribonucleic acid. So this central dogma, the name I am going to say, in the beginning of the class I use the term translation and transcription, right? The same term you can see here, translation and transcription. Okay, transcribe. the message is going to transcribe from DNA to mRNA. So, mRNA is going to carry the message, right? And this mRNA going to translate the message to make protein, and the protein is formed. So, this process we are going to see as transcription and translation, right? So, this is our central dogma. As I said, there are more RNA. Let us see what are the RNA. mRNA, rRNA, tRNA. You can see here. So, this mRNA acting as a messenger, going to carry the message. tRNA is going to translate. Okay, rRNA, ribosomal RNA. You can see the three types of structure, right? Another important question, even NEET based question, make a note. This is the last process where translation, how the message is going to translate. In that type, we are going to see the rRNA is the largest RNA they are saying, which consists of RNA and protein. Okay, and it's the largest RNA, as I said, it's a 1 mark question, make a note. It helps in protein synthesis. Okay, I'll just highlight which is the largest RNA, 1 mark based question. And you can see the ribosomal unit. Already we read in the molecular basis lesson, smaller subunit, larger subunit, right? So, totally I have introduced about the DNA. So, what are the types of RNA, and then we saw about the structure, this DNA, how it is made up of, the nucleotide, polynucleotide structure. In next class, we will see in detail about something here we are going to see. So, what is this? Have a guess. Thank you.